and the group question will be displayed. Now, have you seen live? Live. Click on live. Click on live. You see what will be displayed.
Yeah,
Live. Click on live.
yes.
Okay. Let's see. Comment as soon as we are getting there. Make comments. So we have group task is already being displayed. Don't forget that the group task, you have to work as a team. Then you will now do the presentation by the next class. Hello. Write your own, or is it not displayed? Write your own. Write the one that pertains to your group. And you are going to make presentation on it. You are going to make presentation on it. So prepare for it for the next class. Once you have written your own, I'm just giving you 5 minutes for this one. Immediately after this one, I'm going to display the pre-class question. So just write only the options. You write it. And I need only your pet name. Only your pet name should appear on it. Okay? So I have 2 minutes more to display this. Two minutes more to display this. Two minutes to display this. I pause. I told you that you should make sure that what is going to change ID of your phone. For me. Happy? Or maybe your name appear, or the name of your phone appear there.
No, Wi-Fi.
Just give me. Hello, you have to work according to this. Elijah, why you standing? Why you have joined another group? So that means okay. Are you through with copying your group task. Okay, so the next thing that should appear now should be the pre-class. The pre-class test. The pre-class test is going to be displayed now. So the pre-class test is going to be displayed. It's already been displayed. Have you seen it? It will come. It will come. It will appear now. So you are going to do both pre-class and post-class. The pre-class, you will do it now. Then the post-class will be after I might have done the presentation. Then another quiz is already waiting for you. Has it displayed?
Yes, sir.
Okay. You are expected to write the option. If A is the correct option, write A. What is in front of that A? Write it. No, write it. Tear a sheet of paper and write it. And I only want to see your pet name. That's why we have the examiners. They are already sitting there. They will mark it and they will know. The examiners, they are the ones sitting down there. That should not cause any noise at all. is already displaying. 4 to 6 is already displaying. After some time now, I'm going to four to six is displaying already. Am I not right? I mean, am I right? Yes. Okay. Ẹ má dá wọn lóhùn. Àṣírí yín máa tún sìn. So 7 to 10 is already coming up. Seven to 10 is already displayed. It will come up. Can you see 7 to 10 now? So please submit. I want you to look at what is displayed for you now, and I want you to focus it, pay attention to it. Any group, listen, any group that is able to explain it to me is going to be bonus mark for that group. So pay close attention to it. What is displayed on your screen now? The digital page of life sciences. So look at it with close attention, study each component, and you should be able to explain it. Any group that gets the correct explanation, that means it's going to be a plus mark for that. It's going to be a bonus mark for that group. Study it. Study it. You are free to study it together as a group, because it's going to be for the whole group. So study it. Who can explain the infographic that is displayed? Bioinformatics as the digital bridge of life sciences. Digital bridge of all life sciences. Now, one other advantage of you participating in this course, if you are offering anything you do in bioinformatics already, that is artificial intelligence. So what we are trying to say is that there's no way you can do bioinformatics without doing what is called artificial intelligence and machine learning, because most of the data will be run by the use of machine learning. Now, let me try and explain key features of the infographics. Now, if you look at your left, you are going to see where they talk about blasts. So, as a bioinformatician, some of you that were around in the first two classes we had, maybe 2 weeks, so you were able to do blasts successfully. And I said, as a bioinformatician, the first thing that you must know is to know how to perform what is called blasts. If you say you are a bioinformatician without knowing how to do blasts, that means you have not started. You don't have any foundation at all. And I told you also that blast, why do we need to blast? I won't be able to go back into everything that has to do with blast, because in that video, in that class, we were able to do what blast was, what blasts were, what are the interpretation of blast, what are the biological meaning or interpretation that you can make from blast. So I've done that for you. So I said, under that, you are going to have what is called a query sequence. The sequence you have at hand is called the query sequence. And the sequence that you have all over. Now, I want you to look at where you have the query sequence. Look at this side of, look at the color. Look at the color of the query sequence. When you look at the other side, you have the color matching. That means you have what is called, if you look at this place very, very well, that means you have an isolate, you have an organism already. For example, I told you that day that one of the reasons why we must do blast is for diagnostic purpose. For example, if somebody is sick now, gone were those days that you have to go and culture. You go to the laboratory, culture, and at the end of the day, you just see that the isolate that you are able to get from is still going to be an isolate or properties that at the end of the day, you might not even be able to diagnose it correctly. So for you is to be able to get your isolate, then you pass it through the bridge. If you see that bridge, you are crossing over to the other side. That bridge now, you now have the world that is the global database. I told you the UniProt. UniProt is for protein. The gene bank is for DNA. Okay? And the one you have in the middle. So if you look at those lines, you will see that that yellow enters into the first box, which is the middle one. Then those ones that are related to protein, you have it compared with that one. Then that of the DNA or nucleotide is what you use the gene bank for. And by the time you now come out at the end of the day, you now compare with the digital libraries, which are gene bank and UniProt. So then the next thing, I told you one of the previous presentation that when you have DNA, structure might not actually matter. But when you now have protein, that means the folding, the way it is folded will go a long way as to determine what is going to be the function of that same protein. And in the year 2000, 2020, 2020, that was when they were able to make use of a kind of a software that they call it AlphaFold. So that AlphaFold, they use what is called artificial intelligence. And that artificial intelligence now make use of compare more than 20 million proteins in terms of their structure. And at the end of the day, you pass it through that, and you have the protein already there. Okay? So that is that. Now, having said that again, I told you again that from that diagram, you are going to see you are already seeing what are the foundational biological databases, the DNA, the sequence type, you have the genetic. So that means your sequences, your source sequences could be from DNA, it can be from RNA, and it can be from protein. So now depending, when you now look at this other side, this other one side, all the blasting we are doing, that is comparison, all the sequences that we have, all the structural comparison that we were able to use artificial intelligence, that is like for AlphaFold, to interpret the structure, all of them will now be useful in real life application. And that is why under the real life application, you are able to do the exact pathogen identification. After you must have subjected it to bioinformatics procedure, that is when you'll be able to diagnose correctly the exact pathogen, whether it is a bacteria, whether it is a virus, whatever it is. Then apart from that, the real application of bioinformatics is also talking about personalized medicine and drug discovery. Personalized medicine and drug discovery. Then the third one is refers fascinology, and that is what is called the infographics. The infographics. if you now have the digital code of life displayed for you, let me just signify. Has it displayed? Okay. Now I want you to look at what is in the central. What is in that central is that the definition of bioinformatics, which is the science of collecting, storing, analyzing, and interpreting large volumes of biological data using what? Computers, algorithms, and statistical tools to understand life at the molecular level. So that is everything that has to do with. So that means when you are going in your journey of bioinformatics, there will be a time that even you yourself, you'll be asking yourself, am I a chemist? Because what you will be learning in biology is going to be like if you are still a chemist. And that is when you will be talking about drug to drug interaction. Because bioinformatics nowadays has gotten to a level whereby you will just use computers and you will now combine different structures, different molecules or macromolecules that they have drug potential. Gone were those days that people would just go and get plants and they will go and collect almost all the plants in the whole of the whole world, and at the end of the day, even if they use it to produce drugs, at the end of the day it might not be so effective. So before they will be able to get just one drug, in those days it can take them even more than 20 years for them to come and know or discover the drug it is going to be made for. But nowadays, because of our knowledge in bioinformatician, in fact, I have over the past 2-3 years now, I have 4 groups in my project group. Any student that is going to come under me will work in any of these 4. Number one, we have people that are going to work in the laboratory. Those ones, they will work as wet lab scientists. That is, they will collect sample, they will isolate, they will culture, they will use plate, they will use a lot of physical, physiological differentiation, and at the end of the day, they will just come up with, and so that is wet lab group. Another group, I have them as drug group, drug discovery group. This one, they will work. The 3 other groups, they are bioinformatician. So what they will do, in fact, the last class, I think last year, that was when I introduced the drug design group. And honestly, let me tell you one thing. That was the first time I would be doing something about drugs. In fact, only 2, Zainab and I think Aisha, those 2 coordinated the whole group. I just gave them, okay, you are going to do design drug. And at the end of the day, come and see the excellent result they came up with. So that is a drug design. So what you are saying is that sometimes as bioinformatician, you'll be thinking that, oh, are we into chemistry? So because that has to do with a drug design. Then at a point in time, there will be a molecular biology level whereby as bioinformatician, you are going to have knowledge. You must know. I've just mentioned DNA, RNA, and proteins. I've just mentioned what is called the central dogma theory. All these are very relevant in molecular biology. Then there will be a time whereby people will even think of you as, are you a medical scientist or are you a microbiologist? Because you are going to define the exact targets and microbial community. Under normal circumstances, nowadays now, do you know that you can just get a grain, you can just pick a grain of sand and you just apply what is called proteomics. It's also a branch of bioinformatics, and you apply that knowledge and you will be able to know all the microbial community in that group with just one search. So that is the other side. Then there will be an aspect that we will just say, are we into computer science? Because the use of computer to analyze biological data itself is what is called bioinformatics. And some parts, for example, by now you know what is called the e-values and the interpretation of e-values. All these are statistical tools. That is why we call it the digital. Bioinformatics is now the digital code of life. Okay? Now, you can now see some people are now beginning to ask questions. What are the differences between bioinformatics and computational biology? Two of them, they have where they intersect and they have where they do what? They also disagree. And that's why you see them being represented in two Venn diagrams. Can you see it? What combines two of them is that two of them are genomics and system biology. Bioinformatics is a tool builder, because you focus primarily on development of algorithm, databases, and software for biological data management and sequence analysis. But as a computational biologist, there will be a time that you'll be building a model. And that is when artificial intelligence and machine learning is going to come into place. So that is the area where bioinformatics and computational biology, where they are actually related. Okay. Now, the milestones in time, milestone timeline. What do we mean by milestones? Where are we coming from? Is it that bioinformatics, is it just starting now? Or when is it actually, when has it actually started? So, and that's why you have that long journey. That's why you have that long journey where you have 1953. The whole journey of bioinformatics started by Watson and Crick. When they discover the structure, the exact structure of DNA, which is the double helix. You know, I told you at the beginning of this presentation that you should understand the structure of DNA, understand the component of DNA, see it as where your journey must actually start from. If you are going to be a bioinformatician, if you are going to be a molecular biologist, whatever you are going to be in biology, the journey actually started in 1953 by Watson and Crick. And in 1965, that was when the first protein sequence database was created. That was done by Margaret Dayo in 1965. Then the journey also continued until 1990. That was when the Human Genome Project actually started, and the first time that BLAST algorithm was being launched. What do we call the Human Genome Project? The Human Genome Project was an attempt to be able to sequence the whole human and come up with all the alphabets. You get the whole human and you sequence it, you synchronize it into all the alphabet that is made, that human is actually made of. That was when, so that journey started from 1990. And in the year 2001, that was when the first draft of Human Genome Project started and was published. And they now said that the whole of human, as you are seated, as you are looking at me, as I am even presenting now, that I am made up of more than around 3 billions base pairs. That is, those base pairs that you now have guanine, cytosine, adenine, and this. All of them, when you count them together, they are around 3 billion base pairs. That was when there was now a problem. In fact, let me just tell you one thing. When I was my project, I just sequenced Mycobacterium tuberculosis, which is just about 4.5 million bases. When you compare 5 billion, how many 5 billion can you get from 3 billion? You can see that human, so that means you can combine TB more than even 1 million of it before you be able to even get 1 billion. And human is made up of 3. So that was when there was now a problem. The problem was now this large database, this large data that they have been able to sequence, how would they utilize it? How would they analyze it? How would they interpret it? That was when they now started another journey. The first problem was how many base pair are we made of? And we started from 1990 till 2001. And that one cost more than 3 billion US dollar to sequence human. Then, now, they now said, the whole world now came together. They now said, okay, if we are able to make use of more than 3 billion to sequence just one person, how many billions is it going to take to sequence the whole population? It's going to take a lot. And that was when they now came up with an advice and a kind of another research that, number one, they want to make sequencing to be accessible. That by now, the plan was, or the goal was, that you should be able to just go to bioinformatics lab, just give them your own sample and ask them sequence all the nucleotides in me. And they now said it should not be more than just few billions. In fact, now just the one I told you, I used 212 US dollar per one isolate of Mycobacterium. 212. That was last year. 212, and I sequenced. I think I sequenced 10. 10 because I collected sample from Ondo, Ekiti, Oyo State, Kwara. I collected people that had multi-drug resistant TB. The isolates, I sequenced them. And with 212, I was able to sequence one. Now, what am I trying to say? The journey will not stop there. There will be a point. They want us to get to the level that if you want to sequence the whole, not even bacteria, that you should not spend more than $20 to sequence the whole to a whole human. So the journey has not started. Then, in the year 2001, if I wanted to sequence the same organism as at that time, I was going to spend more than 3 million to sequence one. But last year, I was able to use 212 to sequence it. That means we are already getting into that place. Although we have not reached into that level, but we are moving there. And why are we going into that? One of the reasons we are going into this is what we call personalized medicine, personalized diagnosis, personalized pharmaceutical or personalized drug. So that if you are able to have my sequence, you should be able to go into your laboratory, go and design the drug that is going to be on me and me only. Gone were those days, the whole paracetamol that is going to be made in United States is going to be made for the whole world. And at the end of the day, it will not be able to even cure your headache, so to say. So what they are now looking for now is what is called personalized medicine, personalized diagnosis, because the bacteria that is in hay might be quite different from. So it's going to require different drugs for you to be able to kill the bacteria in you than what is going to be meant for B. So that was when they now came up with, in 2005, what is called the next generation sequencing, which drastically reduced the costs. Just like I told you, it was from year 2005 that the drug. So, and by the year 2020, I have just mentioned That was when they now integrated what is called artificial intelligence into bioinformatics. And what did that artificial intelligence, what did it do? They use a structure, a database that is called AlphaFold to determine the structure of more than 200 million proteins, including proteins that are very, very essential for I and you. And that was 2020. And that's the reason why, and that is the point at which we are now. Don't be surprised, and I've just mentioned it to you, that you have this institution will be one of the few that are offering bioinformatics as a course. No other polytechnic in the history of this country that is offering. Go and ask very well. So what we are saying in essence is just that the mind is for us to be able to have people, bioinformaticians, that will now key into that and be able to make use of artificial intelligence to be able to do a lot of analysis. Okay, so this is year 2026. Now, what I just mentioned to you, there is what we call the metagenomics. Has it been displayed? Metagenomics. Metagenomics is just talking about analysis of, you want to know the totality of microorganisms in your environment. And what will you do? You make use of proteomics, which is the study of protein. You make use of transcriptomics, which is RNA, and you make use of genomics, which is that. We won't be able to dwell too much on this slide because it is a bit higher and advanced. Now, the next thing is sequence alignment. For those people that were in the first two classes, I mentioned and I explained what is called alignment for them. Alignment is bringing two structures together to be able to see where they are different and where they are the same. And when you look at the basis, the sequences that is being displayed there, you will see that G in sequence 1, in sequence 2, there's a gap. Those of you that were around, what is the score of gap? How will you score gap? Huh? That were around that time. How will you score gap? What's the score of gap? Huh? What's the score of gap? Talk now. What is the score of gap? I told you. Elijah, what's the score of gap? Gap. Huh? Score of gap. I'm asking those that are around. Elijah, you are there now. What's the score of gap? Elijah, you are there too. What's the score of gap? You are there. What's the score of gap? Huh? What's the score of gap? Score of gap. How will you score gap? How will computer? What is going to appear on computer if you have a gap? Hmm? You just wrote it down. You are not reading your notes. You are not watching the videos. What's the score of gap? I gave you two sequences that day. And I told you where you have similarity, what's going to be the score. Where you have differences, what's going to be the score. Where you have gap, any omission, what's going to be the score. Wherever you have similarity, that is +1. Wherever you have gap, that is -2. Wherever you have the similarity, that's differences, it's going to be -1. Okay. Now, even in that place, in that same presentation, I also mentioned the differences between pairwise alignment and multiple alignment. So go and read that when you go on. Then The other one is when I gave you structural bioinformatics, that I gave you um, proteins, then you pass it through computer, that is artificial intelligence, to interpret it to you. So you will see it in the presentation. Don't forget that immediately after this presentation, we are going to do the quiz together. Okay. The global data repositories, that is, what all those data, all of these data, where can you find them? Anything that has to do with nucleotide, you go to where we call the gene bank or NCBI, or you go to assemble, that is nucleotide. That is where you can, where information about DNA is being stored, or nucleotide is being stored. Then, if you are looking for protein and the structure of protein, you go to where we have uniprot / sweetprot. We are going to see all this. Then PDB. Anytime you want to learn about a metabolic and interaction pathway, you go to a database that is called KEGG. So if you want to understand about Krebs cycle, glycolysis, and some other, you make use of that. And where you want to talk about microbial metagenomics, so you make use of Silver and MGRAST, okay? So bioinformatics toolkits, that is, as a bioinformatician, what should be in your pocket every time? What should you be coming with every time you are coming for this class or anything in bioinformatics? Number one, you must know how to use BLAST. You must know how to use cluster omega very, very well. You must know how to use Mosul very, very well. You must be able to know how to use the MAFTs, that is multiple sequence alignment tools. You should be able to make use of them. Then another thing is assembly and annotation. You should be able to know it. How can you assemble? Because immediately after you must have gotten all the sequences, how can you bring them together and assemble them, and you'll be able to know that, okay, this protein, when you combine all this is for this function. That is assembly and annotation. Then the phylogenetic tree, you should know very, very well. Then scripting language. That is when you need Python, you need the pandas, you need R. For example, in bioconductors, all these, the last one is a kind of advanced in bioinformatics. So you should know, you will come across the BLAST selection matrix. You come across BLAST N, that is talking about nucleotide. BLAST P, that is talking about protein. BLAST X, that is talking about protein as well, but it has to be translated nucleotide. If you come across T BLAST N, that is talking about protein and translated nucleotide. Then you can also come across T BLAST X, which is translated nucleotide, and the database is going to be that. Then you have to be acquainted with e-value. The closer you are approaching zero, that is talking about very good alignment for you. And you also need the other matrix, which are going to be secondary, but the primary is e-value, which is very, very important for you. So application of bioinformatics in transforming microbiology, and this, you will know them. Then application in clinical science and drug discovery, I've also mentioned that. Now, what are the ethical genome? Okay, the ethical genome, you will see some of you that came for the AI training day before yesterday. I think they told you about the ethical use, the ethical use of AI. That is, thou shalt not steal, thou shalt not, you must respect another person's privacy. So it's also very, very similar to this. Privacy is there. You must seek the person's consent. Under normal circumstances, I want to collect sample from you, and that is where the journey will start. If I want to collect, even if I want to diagnose or even I want to treat you and I want to collect your information, I must get your consent. Consent, it must be there. So, and having done that, bioinformatics is already talking about application in clinical roles, computational roles, and at the end of the day, it is very, very important and very, very useful for us as a bioinformatician. So, having said that, I want to display the quiz now. The quiz now, that is post this. So let me give you 1 minute to go through the presentation again before I display it. The one you had before the class is pre-class. That is before you are able to listen to this. Even if you fail that, it might not be too necessary. But this one, if you now fail even after the class, that is when we know that you actually need a surgeon. You need somebody that is that means you have a problem understanding. The question is displayed already. The quiz. Okay, this is what we do. This one post. Do it in the comment section so that the whole world can. Do it in the comment section. So So, are you done with number one? That's the option. Hmm? That's the option. Now, comment section. Comment section. Your name is already. Okay, add your pet name to the answer. As you are submitting, add your pet name. Only the pet name. Are you through with this? Yes, sir. Yes, sir. Okay, I'm going to each should be displayed for only 2 minutes, maximum of 2 minutes. So which has lapsed. So let's go to the next. The next question has been displayed. Second question is coming up now. As question 2 displayed. Seconds more for number. Comment, please. Comment. So that's in order to enrich the algorithm. Comment, like, and show love. Comment, likes, and show love. Number 3 question is already coming up. Question 3 is already displayed. How many of you have seen it? Question 3. Uh-huh. Okay. It's going to be on for 2 minutes. Question 3 is already displayed. Done. Okay. Question 4 is displayed. It's going to come up now. Uh, as number 4 displayed. Let me know if it has displayed. Refresh your internet. How far? Huh? Okay. So enjoy it for the next 1 minute. Okay. Number 5 is displayed. It's going to come up now. As number 5 displayed. Displayed. Okay. Okay. Question 5 saying, okay, let's go to the next one. We're already in question 6 now. So watch as it will be displayed. Cannot comment. Cannot commenting. You do not share comment. You do not buy. I want you to waste like on this. Waste comments. Comment section. Has it been displayed? Yes, sir. Okay. Number 6. Okay. So that means we have 4 to go. Hmm? Question 6, Abby. As you go to number 7. Okay. Yes. You see, number 7 is already there. It's going to come up now. Number 7. Number 7, has it been displayed? Okay. So enjoy number 7 while it lasts. Comment, like, and share. Question 8 has been displayed. Question 8 has been displayed. It's going to come up. You are not to complain about the question. Just do it. Don't complain about the question. The question was generated with respect to the lecture you just listened to. Yes, do you have number 8 displayed?
No, sir. Yes, sir.
Oh.
Yes, sir. Yes, sir. Yes, sir.
So number 8 is already displayed. I'm going to display number 9 now. Number 9. Should I?
Yes, sir.
Are you sure?
Yes, sir.
Okay. Okay. Number 9 is displayed already. Have you seen it? He said yes. Don't forget to like, share, and comment. I should display number 10,
Yes, sir.
which is going to be the last one. Ara oko. Okay. Submit. Submit. If you have submitted, let me see your hands up. If you have submitted, submit. Come and see it. Come and bring it. Let me see. Submit there now. Comments.
Submit. Press submit.
In the comment section, go.
Okay, we can submit.
After you must have submitted online, it must have graded you now. When you submit, has it graded you?
No. Yes, sir. Comment section.
Okay, yes.
Comment section.
Yeah. Submit, oh, have you?
Yes, sir.
Have you?
Yes, sir.
Mhm. So now look at, it's because you now look at the right answer now. Look at the answer now. It's going to be displayed now. Look at the answer now. Have you seen it?
No, sir.
Number 1 is A. Huh? A. Ọgbẹ́ kí ni wá? Ó ti gbélé ìwá náà. Have you seen it? Okay. Now let's go to number 2. Number 2. Have you seen it? Number 2. Have you seen it? Number 2, has it displayed? Check number 2 now. Once you get to number 2, let me know. Who can tell me the correct answer to number 2? Okay. So that's B. Let's go to the next one. Let's go to the next one. Once number 3, correct answer to number 3 has been displayed, let me know. Has number 3 been displayed?
Yes.
Have you seen number 3?
Yes. Yes.
So number 4 is coming up. Is Man U playing today? Okay. Once number 4 has displayed, let me know.
I saw it.
Has it displayed?
Yes.
So let's go to number 5. Number 5 has been displayed. It's going to come up. It's because I didn't answer anyone. That's why it's bringing skipped. Why I didn't answer. Oh, God. If you have seen number 5, let me know. Have you seen it?
Yes.
What's in here?
Okay.
Okay. Go to number 7. Okay, number 6. So number 6 is already coming up. If you have seen number 6, just let me know. Number 6. Who else has seen number 6?
Yes.
Okay. Let me go to the next number. Once you see number 7, let me know.
X.
Who else has seen number 8? I mean number 7.
That's it.
Who again? You've seen it. So let's go to the next one. Once you have number 8, let me know.
G.
If you have seen number 8, let me know.
S.
Who again? Have you?
S.
Is that number 7 or number 8?
Number 8.
So let's go to number 9 now. Once you see number 9, let me know. Let me see. Once you are in number 9, let me know. Number 9. Who else? Who else? Okay, so let's go to the last one, which is 10.